Vamos conversar, porque senão o horário começa a atrasar as outras mesas. Bom dia a todos. Daniel e Carol, super obrigada por me convidar para moderar essa mesa, que para mim é bastante importante. Então, uma mesa de entomologia que está muito bem composta. A gente vai ter desde o status de controle vetorial, quanto processos que moldam populações de niçorrincos da linge e estudos experimentais com
And
vetores.
experimental studies with vectors with Dr. Paulo Pimenta at the end. We're going to start with Dr. Allan Kardec.
Bom, então o doutor Alan já pode vir subindo, então.
Dr. Allan, you can come up stage.
É do Instituto de Pesquisa e Ciências Tecnológicas do Estado do Amapá.
Allan Kardec Ribeiro Galada from the Institute of Sciences of the State of Amapá, where he coordinates the Laboratory of Medical Anthology. He is a doctor on parasites at the Federal University of Pará, and he has a lot of experience. I would like to say that he is also a great collaborator of our program in partnership with Dr. José Bento Lima. Dr. Alain is going to make the lecture Vector Control in Malaria and Transmission Areas. The next 20 minutes are yours, and we let you know when there's 5 minutes left. Good morning. Thank you, Simone. Thank you, everyone, for the invitation to present our paper today. We wanted to assess the vector control based on entomological data. We're going to talk a little bit about this, and then we're going to do a brief reflection of how our control is in Brazil.
Eu fiz questão de buscar essas informações um pouquinho no passado, em relação a como é que começou o programa de malária no Brasil, né?
Before I start on the topic, I would like to talk about how the control program started in Brazil. It's important to remember that at the time, the basis of the vector control was exactly what we do today. We used insecticides that everybody knows, our sprays that everybody uses till today, and we got this basis from the Rockefeller Foundation, the Deneru Manual, and we did this to the cities. And then we eliminated the mosquito birthing sites. In 1939, we see that this control methodology had been used for a long time already. When we look at today the manual of the Ministry of Health, there's a note of the Ministry of Health saying that to control the vectors, you need entomological information. So what I had at Deneru, what I had at the Rockefeller Foundation, which was the study of entomology, was basically retired. We know very few groups that do field entomology, because one thing is to do entomology in the laboratory, but operational entomology is not a very common practice today. What we had in the past, we don't have today, which is that strong study in the area of entomology. And when we talk about control, everything that we need in the area of vectorial control, I need to study parity, if a certain insect is resistant to insecticides or not. To do studies, we need to do control, we need study. So when I go to table 2, I see that I need to have this information, that necessary, the N information, so I can have control. If I spray a house, if I hand out mosquito nets, I need entomological information, which gives me the basis to do the control. And why is that? I'm going to get a very simple example. If the peak of a mosquito that transmits malaria is at 7 p.m. and that residents they go to bed at 10 p.m., they are not under the mosquito net at 7 p.m., which is the highest activity of the mosquito. There are areas where I hand out nets and I spray houses and it works, but other areas it doesn't work because it's a leap in the dark. I could be reaching the target or not. So when I talk about gradual uh, elimination, I need to understand if the mosquito is going to feed inside the house or not. Because if they're not going to feed inside the house, the spraying is not going to make sense. So is the mosquito feeding inside the house? Is it having contact with the residents? I have to evaluate that also for the control of the birthing sites and for the spatial control with fog. We can prove technically that it works with entomological basis. And applying insecticides without entomological data is a leap in the dark, a leap of faith. Roberto actually helped me with this information, showing how much the entomological study is going to help us with control. There's an interesting work doing in the Yanomami, being done in the Yanomami territory. There was a technical note about it. They put together the entomology. So in the Yanomami territory, they have 10 people that study entomology. They don't do a control on the territory without entomological studies, and we start having a few results from that. They did all of the studies in laboratory and in the field, and based on that, they defined what to do in each specific area. So it's not a single solution. You study the area, and after you study the area, you do the intervention in that area. And with that, we have a few results of malaria reduction in the Yanomami territory. There was a 31% reduction from one year to the other. It is low, but it's an indigenous land. It's always difficult to do controls. And another example from the gold extraction mine of Gaivota. We did a social environmental study to know if the residents were truly sleeping under the nets, what time they slept. We did an entomological study before we handed out the mosquito nets. All of these yellow points are registered for having received mosquito nets. And then we did a whole work, and before the intervention and after the intervention, we had 94% of reduction of malaria in the gold mining areas, showing that the study is worth it. You have to study the community to do the control. We discussed till this day, myself, Bento, Ana Paula, what truly did that? What truly made it reduce? First, education and health, because we visited the residents every 3 months. We would change the mosquito nets if it was damaged. We incentivized the washing of this mosquito net, so we were able to work with the community for a long period with education and health. And here, we stopped working with them in 2024, and you could see how the malaria numbers started increasing, showing that the intervention, which was not unique, uh, it- but it was only the mosquito nets. We didn't spray the houses or anything else. But after not stopping to visit them, after we stopped visiting the residents, you could see that the malaria numbers increased. So the activity is very important. I got two papers here from Pará. The municipality of Anajás always had a lot of malaria, so we started doing entomological studies before we started doing uh, control intervention. So we got three sites, Santa Luzia, Saituba, and Santo Domingo, where we have the red arrow is where we did the entomological study, and then the control actions based on the entomological studies. In Santa Luzia, there was a reduction of 68%, in Açaituba, 77%, and in São Domingos, 86%, based on the entomological information. Now, another project of Jacaracanga in an area that is critical for malaria, I wanted to bring here two examples. In the village Estirão das Cobras, there was an intervention. There was an entomological study in August, and then there was an intervention in the following months. In the Cató village, there was an entomological study in August, but malaria continued. So, does the entomological study not work? I'm going to explain why. After the entomological studies here, we had the control intervention, and we saw in the Estirão das Cobras village a reduction of 77%. In the Cató village, there was only a 24% reduction. But why? Because in the Cató village, the intervention happened 4 months after the entomological study, showing that the entomological study is not static, it's not idle. We talk to entomologists in the field. We have several different stories, and they're always very close to the community. When you're in an area with the transmission of the disease, you do an entomological study and then an intervention right next, you have results. If you go 2 kilometers away, you do an entomological study, and the results are totally different from what you found 2 kilometers back. We cannot say that the mosquito stings at uh, 7 p.m. We collected mosquitoes at all times, at noon, at midnight, at 2 a.m., with a high peak of infection and density. So any control that I do outside this time will not have impact. That is why it's important to do an entomological study. There's another very interesting work here. Gilberto helped us with the information for this paper. Today, in the system Vetores Malaria, you can enter all of the survey that is done, and you can cross-reference with cases of malaria and intervention methodologies. So we got two examples here. This is where the malaria cases are happening for density, and that's where we had an embedded mosquito net installed. So there is malaria, but there's no mosquito nets. Or here there's zero malaria, and there's a lot of mosquito nets. We get also here in this image the cases of malaria and the spraying of houses and delivery and installation of mosquito nets. And gathering what we have in terms of strategy today, if you remember the first slide that I was talking about, the Danaroo and the Rockefeller Foundation, basically for control, we have everything that we used to do then. We did not add anything else the um, other than this insecticide-treated bed net. At the time, we had at least spraying and the treatment of mosquito birthing sites. Today, we have spraying and insecticide-treated bed nets. We don't do a lot of activities in control of birthing sites. We are below what we did 80 years ago. We have a lot of studies being done to improve and to diversify our methods. So from 1939 to 2026, what we have done is the indoor residual spraying. Basically, when we talk about control of vectors, that's basically it. So we're not saying that it doesn't work. We're not saying that it is not functional, but we need to think a little bit. First, when you think entomologically, when you get to an area, you have a lot of question marks, a lot of questions. What can I do here? But for me to do something in this area, I have to study it. I have to get to know it. I have to get to know the vectors. So AI is fabulous. They just added the wrong mosquito. It is not this mosquito, but I was trying to make this image on AI just to illustrate it to you. So we wonder, what do we do? When we work in the field, you control a mosquito in this area and you cannot control it in the other area. They're doing the same activity that you have been doing. So I got a work that was published by our group. It is an old study, but we should reflect upon it. In 2004, there was a work done in Macapá with 3 riverside communities. And what we noticed there is that every day at 6 a.m. for 1 hour, we did uh, capture resting capture inside the house and in the vegetation around the house. We noticed that 72% of the ingurgitated mosquitoes do not rest inside the house. So I go back to my question. If they don't rest inside the house, should we do indoor spraying, indoor residual spraying? We need to study that. But as I said, entomology is not static. In the same area of São Raimundo de São João, not even a kilometer away, I have more mosquitoes inside the house than on the vegetation. So maybe it will work in São João, but not in São Raimundo. That is a question. We need to observe these details. Something else that's very important in the same work that we did, we always say that the mosquito is anthropophilic, as if they did not feed on anything else other than humans. But you can see here, bovine, 60%, almost 60%. They need blood. We found mosquitoes with human blood, bovine blood, aviary blood, rodent blood. So we need to study. We need to understand a little bit more about this mosquito. We think that we already have a lot of information on entomology, but we don't. I assure you that we don't. We still need a lot of study for vectorial control. And two initial questions. What is the question that I asked myself today? IRS, the indoor residual spraying, does it work? I guarantee it works to kill mosquitoes. We do a lot of tests in Macapá. This is our test house where we test a lot of different types of surfaces. And there are products in the market that have residual effect for 12 months depending on the surface. So I do have a good product. I know that I have an insecticide that is enough to kill that mosquito. I know it. But the question is, because here I place a mosquito inside a cone exposed to a wall with insecticides, but in the community, in the place where people catch malaria, is this effect the same? Do I have this effect in the community for malaria reduction? That is what we have to ask ourselves. Also, according to the data of the manual that we have, we always say that I needed coverage of 80% of the houses so that it can have an effect on the malaria control. I don't know any municipality in Brazil today that can spray 80% of its houses. Should we maybe reassess this percentage? Could it be a lower percentage? Or maybe what we're doing is not working? We should think about reassessing it when we talk about control. Another issue is how many mosquitoes fly inside the house a day? What is the percentage of these mosquitoes that are exposed to light inside homes? Gilberto collected in Macapá over 300 insects, over 300 mosquitoes. One was resting and exposed to light inside the home. We have to assess that. We have to do an entomological study to know if there's actually an impact in the transmission of the disease and the mosquito control. And another question that I asked myself, and today I think we're going to answer the questions here. It's only the Anopheles narynx that transmits malaria. I did an entomological study in an area that did not have any of these mosquitoes, and there was transmission of malaria. There wasn't a larynx, but there was malaria. In my doctor's degree, I studied other species that transmit malaria, and there were species that transmitted 3 times more malaria than the larynx. When we say let's control, we say enough is a range. And we have to think more about that. And thinking about vector control, does space-based automation work? Does fog work? I don't know if it works because when we do fog, when we do spatial application, we do we do this procedure in the work at the time that the people are arriving home, not the mosquito. It is not a methodology without residual effect. Don't read, if I don't do it the correct time that the mosquito is searching for blood in the residence, it's not going to work. A study by Dr. Bento is going to talk about this that we carried out in Manaus with entomological data that shows that it is effective. It does work. It reduces mosquito parity, which is what it's supposed to do. But we have an insecticide in the fog that it's about 500 years that it's been there. It's 500 years, it's an exaggeration, but it's the same product. Since we don't recommend, we don't search for new chemicals. And another question to conclude. Does larval control work? Denero used to do that, Sukan used to do that, and we almost don't work with the larva control today. What do we have in the market today um, to do control? I included here some new methodologies. There's a series of new methodologies, series of new methods, and we're not looking for information on that. So we need to stop and forget trying to think that we know how to do control and go back to the past of the entomological studies, because only the entomological studies are going to give me a good control. Thank you.
Obrigada, professor.
Thank you, Professor.
Eu chamo agora o professor José Bento Pereira Lima.
I invite now Professor José Bento Pereira Lima from Fiocruz, Rio de Janeiro, with the title of the lecture, The Contribution of Vector Control for the Elimination of Malaria. Professor José Bento Pereira is a full professor in the surveillance of insects and the disease of IOC. He's also an entomologist, and he works especially with Aedes and Anopheles conicides, working in the surveillance, control, and resistance. He's also a member of the program of post-graduation in tropical medicine of IOC and the technical committee of advisory of the National Program of Prevention and Control of Malaria and the Institutional Research Committee. Thank you very much for your participation, Professor.
Bom dia a todos e todas.
Good morning, everyone. We will talk a little, actually, as she introduced me, I am José Bento, I'm from Fiocruz, Rio de Janeiro, and we're going to talk about the contribution of vectoral control, thinking about the elimination of malaria. Actually, I brought here a few studies that we have been recently developing in the sense of trying to achieve control. Just to contextualize, as Alan said, our malaria is still concentrated, most of the cases, in the Brazilian Amazon, where we have 99.9% of the national cases. The primary vector is still Anopheles darlangi, because we don't have studies showing the involvement of other vectors. We have a few evidence, but not studies. And for control, we use BRI, the IRS in English, and the insect-treated bed nets, as Allan Kardec said. I'm going to show you the resistance and the evaluation of the status of resistance of Anopheles laurelingii in the Amazon, the use of G2 interceptor mosquito nets, the use of experimental houses for the study of behavior and evaluation of efficacy of insecticide-treated bat net, the evaluation, the impact of thermonebulization and the transmission of malaria, the evaluation of the chain of custody of insecticides, and the microstratification of epidemiology. When we look at this map of Brazil, you can see that in Brazil, we just have one case here in Acre for the insecticide resistance assessment. It seems like in Brazil, we don't have insecticide resistance assessments. So based on this gap, the work by DESI that is published, from the 2021 to 2024, we collected females of these mosquitoes in 28 locations in the 9 states of the Amazon. The results from that, when we look at the products that are used in the mosquito nets, the bed nets, and in the household application, we can see that from the 19 populations evaluated with deltametrine, only 4 were susceptible to the product. The same thing goes for ethopemprox. Ethopemprox is an insecticide that is now being switched for IRS, indoor residual spraying, and from the 18 populations, only 5 were susceptible. Regarding permethrin, that is used a lot in insecticide-treated bed nets, from 18 populations, 11 were susceptible to these products, that permethrin in this case.
And now Brazil is part of the WHO map showing that we have insecticide resistance in the country. Now we are able to see ourselves in the map. And the other study that Alan mentioned is this new generation bed net, and we evaluated in this mining area in the state of Macapá and in a neighborhood in the city of Coari in the Amazon. And this bed net uh, was used for 3 years. It was a long-term study, and the most important that we've observed so that we could be successful in implementing these strategies is the engagement of the team and the engagement of the population. So with that, we are able to advance and get results. And the results that we have here is a refusal of only 47%, showing the importance of this engagement. And when we see the results, we see it diminished. That's when it started in 2020, and we had a drop of 65%, and it over the time in 23, the 3-year study ended. It wasn't different in Quari. We had a uh, the great drop of malaria cases in the region. So if the tool is used with a good work in terms of education, we are able to get excellent results. So we conclude by saying that it's very important to have education in healthcare, that the tool works and it can be used in the control and elimination of malaria. Here we have a different study, the houses that we built in Quari. The idea was to evaluate in loco how the mosquitoes behave um, when faced with the tools. This is Daniele's doctoral study, and we've been working with that. And this is Quari, an interesting city in the Amazon. Here we see how the houses are behaving. We have 6 houses. We initially evaluated if the mosquitoes were entering the houses, and then we evaluated the insecticide-treated bat net to see the behavior of this mosquito concerning the entrance, the exit, the mortality. rate. And we see here that we have a high mortality considering the insecticide-treated bad net, and in the Interg2 86.9%, and all is SatNet 91.6% of mortality. And here we have the entrance rate, and in the control you see there's no much difference from the control with uh, the insecticide-treated bad net, so it doesn't interfere in the entrance, but a little bit in the exit. And when we observe mortality, you see a high mortality in the 5 houses with the bad net, and little mortality in the case of the houses, the control houses. Here we are talking about fog. It's controversial. People say we shouldn't use, so it's a tool that shouldn't be recommended to everything. As Alan just mentioned, we we should have the entomological study to see if it works. So that was the municipality of Iranduba in the Amazon. We chose two places, one for intervention and the other for control. And then we had an exchange of these two products. So we evaluated the mosquito density, the percentage of female given birth, and the bioefficacy, immediate bioefficacy of the insecticide application, the resistance level, and the infection rate that is being studied by uh, the lab with Claudia Ribeiro. And then we had the intervention at the first place, and then we had this space and the intervention in the second area, and this control area became intervention area, and the first area is here. So when we observe how this was performed, we had collection all the time, 36 nights of collection in all of the places in the application area where you have the red arrows, and in green is the mosquito collection. And here we saw the behavior of the vectors. We had a high collection. Most of the mosquitoes were Anopheles darlingii. The period was uh, the from between from 6 p.m. to 7 p.m. when we applied the insecticide. We also worked with these cages where you put mosquitoes in some of the houses, and after applying the product, we evaluate the mortality after 20 minutes and then in 24 hours to evaluate the efficacy. And when we observe this chart, concerning the validity of the product, this intervention guarantees that it was accomplished because we see mortality in the cages. When we observe the vector density, we observe that there was not a lot of difference. You didn't diminish density. In the control area and the application area, you still have the same density, but when you observe age, which is the most important, you're killing the old female, and new females are coming to the place, showing that the methodology is efficient in its application. And these are some outcomes, especially concerning resistance. When you see that this product that we worked with Iranduba population with the recommended doses, we are only killing 12%. So uh, there's a resistance to the insecticide in the area. And we work with 5 times the doses, we killed 69%. So in this population of Iranduba, which is there's quite a resistance to this product. And here the conclusion showing that the fog can reduce 60% of the uh, females giving birth with the persisting effect of up to 4 weeks, it could generate a meaningful epidemiological impact. And it's important to go on with the monitoring and evaluating all the conventional and new tools for the vector control in Brazil, as Alan just mentioned. Another work that we developed was concerning the custody chain. We know the when it goes, but we don't have answers about when it goes back. So how do we do that on site? How do we apply the product? And we had CVAP malaria data sources, yes, and also INMAT, INPE, and IBGE data. And in Marcela's work, we see that the custody chain presents failures that will compromise the efficiency of the vector control. So the current system is operating in a reactive way. We are trying to put out the fire. We only go when we have cases, and maybe it's too late. So we need to have this predictive planning based on data because we have this very important program that is CVAP malaria, and you're able to get a lot of interesting data from that. And the recommendations is having this integrated system so that we can understand what's really happening. And also we should strengthen the entomological vigilance, surveillance, because we lost that over years. This is a different work, the epidemiological microstratification by Eleilson. It was performed in the city of São Gabriel da Cachoeira from 22 to 24. And what did we want to know? What is the malaria dynamics within the neighborhood of the city? He worked with CVAP malaria data as well, with the georeferencing, and then he put together the microstratification of the neighborhoods to understand how malaria was behaving in those environments. And we have some neighborhoods with malaria all year long with lower density, of course, but they are still contributing. And we ask ourselves, São Gabriel da Cachoeira is a region with a lot of indigenous peoples. We have this flow of coming and going of indigenous persons, and we ask ourselves, is the malaria coming or going? Are they bringing malaria to the city, or are they coming to the city to get their benefits and they return to the village with malaria? We must understand better the flow. And something else that we noticed that is that sometimes we abandon the cities and then we have expansions happening. And this is the moment for when we see the intensification of malaria cases, and we should study that. And there we see that seasonality goes on when we have a lot of rainfall. Normally, the case density is lower, and then when and after the rainfall, you have the intensification in terms of case numbers. So you have an idea about the action period when you need to work to control malaria. Concerning perspectives, what could we do? First, we should have more education because we stopped the trainings. We must have them at the forefront. So, what's our objective? What should the agent do on site? Maybe not becoming an entomologist, but they should know about our lineage, they should know how to collect them, identifying, dissecting it to understand the purity, a simple training that could help them on site. Concerning resistance, we've advanced with the ministry. Uh, we are able to produce a paper insecticide treated paper. We also have the tubes that we were able to get the tubes with the ministry. We also have colored ones showing at where the paper is. We have this automatized insecticide treatment. We have the cones for residual evaluation, and we were able to advance concerning insecticide treated tools, and we could advance on site. Something else that we have in the program that is no longer made is the, the breeding control. Gilberto is going to start this work. He's going to start applying uh, in May. And we want to understand if this drone could be used and what's the cost benefit of that. And we should think about when and where to treat, uh, because we shouldn't apply that everywhere. Where should I use the insecticide and the larvicide? This, these fish tanks spread out in the Amazon, how are they contributing to malaria? Is it worthwhile to treat them all? Or maybe I should use in specific sites. This is a work we are starting now. We are going to start application in May, and we hope to have good outcomes with the ministry concerning this methodology for vector control. We must advance with the new technologies. For instance, sugar beets. Should it be used? In which situations? Maybe in the indigenous villages or the riverside communities. We should think about the evaluations. Now, with the experimental houses, we are able to do that on site to understand how it's working in reality. We also have now the face uh, repellent that is being commercialized in the country, and we must advance with that, understanding if it's going to be successful, if it's worthwhile to add to the control to the control of the ministry. And something else that Alan mentioned is concerning BRI. What do we know about that? Is that used? Is that efficient? And when we talk about BRI, we're thinking about an amount of resources that is huge. What if it's not applicable? We are wasting our money. We could direct the resources to the places where we have the most malaria. When we think about DDT and BRI, we had a different type of home. Today, everyone has a TV, and it's very hard this type of evaluation. So that's what I wanted to talk to you. I would like to finalize with this slide, which is the future is the territory. The health surveillance is not only observation, it's also intervention. You must do that to reach the elimination goals of WHO 2030. It's not enough to treat the cases. We must work articulated with community leaders and in the neighborhoods that we consider invisible. The micro stratification was proven a low-cost tool with high efficacy to direct public policies in São Gabriel da Cachoeira, and it could serve as a model to all Brazilian Amazon. Thank you so much.
Pode. É porque o Alan desceu. É melhor para ver lá. Então, bom, gente, agora a gente vai passar o
Well,
tema sobre o
now
processo que moldam
we're going
populações
to
em
talk about
desenvolvimento
this
tardio.
process concerning the population that is
É um
facing
processo de
Anopheles darlingi.
comportamento e
So it's a process that will shape behavior and vector control according to different contexts. Uh, We are going to listen to Paulo Eduardo Martins Ribola, a doctor in biological sciences from USP, the chair of and biodiversity and statistics, and he's a senior researcher with a lot of production on vectors and consistent work with Anopheles darlingii. He's going to talk about the population-reduced structure and transmission, m- malaria transmission and vector control. Thank you, Simone. Thank you, Danielle and Carol, for the invitation. I'm a fish out of water here because I'm not an entomologist. I'm a geneticist, but the idea is to try and bring a little bit of how genetics and how the work in genetics and genomics could help the control of the vector. Well, Anopheles or Nisorhynchus narlingi, it's a mosquito species that is very interesting. There's a, a broad geographic distribution with a high level of genetic differentiation. The Amazon, the Amazon age is around 50 million years. For 15 million years, this mosquito has been in the Amazon feeding from animals, not necessarily people, and it has had an evolution history that is much greater than when they became vectors. They only became vectors after the Plasmodium came to Brazil, both Falciparum and Vivax. And we must think about the evolution of the vector and how it's connected to the malaria transmission. We don't have a consensus if it's a species, if it's not, but again, it's a very variable group, and I'm going to show you some data about that.
In addition to having a very diverse genetics, it also has a very diverse behavior. Alan shown here populations with that have different behaviors 1 kilometer away from one another. So I think we should understand that the mosquito is diverse, but it is also diverse in other aspects like attitudes and behavior, which is going to um, make malaria transmission more difficult to control. This high behavior plasticity has to do with transmission and time of the stings and time that they go inside the house. There are many different things related to malaria transmission, and I'll show you today. This paper was published not very long ago by the group of Danielle Nisse. They sequenced the genome of several anopheles in the Amazon region, and they built this tree. And the most interesting part of the work, in my standpoint, is this graph here in the bottom. It's showing AST, which is a level of genetic differentiation between the Anopheles collected, and comparing it to Anopheles gambiae and Anopheles canusi from Africa. It is impressive that our vector here in Latin America, Anopheles darlingii, is much more diversified than, for example, Gambia and collusion, which are two different species. So the levels of differentiation, the regional differentiation of the mosquito is a lot bigger than the African mosquitoes. This may have to do with the story of the 100 million years of evolution of this mosquito in the Amazon rainforest, which is a very homogeneous and very vast region. Works with other insects have shown that this is this is a good scenario that takes to this level of differentiation among the populations. What I'm going to show you today is the study, the more localized study in two cities of the in Amazonas, in the Amazon, Manso Lima and Acre, and Barcelos in the state of Amazonas. What we did was we collected the mosquitoes in these two cities and we compared the genomes of these mosquitoes to understand the populational structure of the mosquitoes in these two towns. If we compare it to Daniel's work, he has a sample of Barcelos and a sample of Loreto that might be the most similar one to this town of Manso Lima. Actually, I'm working with mosquitoes from the same genetic cluster that Daniel showed in his work. This is an air view of the two cities. The population is similar. The area is not similar because Barcelos is a huge city, but if we consider only the urban area, they're relatively similar. Within these towns, in Manso Lima, we did collections in 3 points of adults, and in Barcelos in 11 points. Some points are wrong, Simone, I'm sorry. We collected the mosquitoes in 11 different areas of the city of Barcelos. These mosquitoes were taken to Botucatu, where we have our lab, where we did DNA extraction from individual mosquitoes, about 350 mosquitoes from Manso Lima and 130 from Barcelos. We put together the libraries and we sequenced at genome low coverage, at 1 to 2 times coverage, and we did a treatment on this data so to try to input more data from the data that we obtained. Why low coverage? Because we have little money, so we cannot sequence as much as we would like. The technologies are becoming cheaper, and we may sequence with a bigger coverage, but in this case, we didn't have that technology or the funding. One very interesting thing that we observed was this index called the fixation index. We call it FIZ. The populations of Manso Lima had a FIZ of 0.5, and for Barcelos, the FIZ was practically zero. It is the same mosquito, it is the same species of mosquito in the Amazon region with a completely different structure. What do these two figures mean? A phase close to zero, what we found in Barcelos, means a random mating. Uh, they are reproducing without any kind of barrier or substructuring. They're inbreeding. Sorry, but they are panmixia, but a phase above zero, which is 0.5 that we found in Manso Lima, means inbreeding, a deficit of heterozygotes. What we have is a Wallon effect, several subpopulations within the population of Manso Lima. When we analyze the mosquitoes individually, we managed to see exactly that. So here we have, you can imagine this as the main component chart. The closer dots are genetically similar mosquitoes, and dots that are further apart are genetically different mosquitoes. So it is very well structured. The mosquitoes collected in point A have a similar genetics, point B have similar genetics to one another, and point C as well. So these collection points, they are about 1 kilometer away. So I'm saying that I have a structuring uh, in a matter of meters. The female or of the population of Anopheles darlingi, they have a birthing site, they have a house where they can feed, they can eat, and the female will always return to the same birthing site and they will feed. in the surroundings of that birthing site. When I look at Barcelos, I can't see right away a structure. And when I run the tests, I couldn't see a lot of structuring in these populations, which is coherent to the panmixia that I found before. What I can see in these individuals is that they have a certain distribution around the city. So I see that I have mosquitoes collected from one side of the city that are relatively different than mosquitoes that I collected on the other side. But we have to understand that the differentiation is still very, very subtle. The mosquitoes are still reproducing randomly or almost randomly. I took this image uh, from a paper on Ascaris umbricoides. It's a paper that I usually use in my parasitology class, but it represents these two populational structures very well. In Barcelos, we have mosquitoes reproducing randomly, so we generate a relatively homogeneous population. And in Mansulima, you have a lot of structuring, so you generate different mosquitoes when we do a fixation analysis. This is a little extreme because Mansulima is a town with over 300 tanks of fish raising. These tanks that they were birthing sites of Anopheles darlingi. In Barcelos, I have a stream going through the city, crossing the city, and a lot of birthing sites that are connected or close by. So the population of mosquitoes is not structured for this reason. Another interesting thing that Daniel's paper showed us is that when he analyzed the genetics of all of the mosquitoes, he saw a region here in chromosome 2 of low diversity. And when he did this analysis of G1, which is an analysis that shows recent selection sites where you have a high frequency of haplotypes, this region here of chromosome 2 has a peak of G1, meaning that you have a haplotype that is fixated in the population. This region of chromosome 2 refers to a site where you have a lot of cytochromicity, which are enzymes that take resistance to insecticides. So the idea is to have fixated in different populations this G1. We found the same locus of fixation in different populations. We found it in the population of Barcelos. This is the highest peak of chromosome 2, but we don't find it in Mansulima, which doesn't mean that the mosquitoes in Mansulima are sensitive to insecticide, but the populational structure of the city does not lead to the fixation of a haplotype in all the population because you have subpopulations. So the spraying of that spreading of that is very small. We would have to have several events so it could spray to all of the different populations. In Mansulima, we have high structured populations, pretty much because of the fish tanks for pisciculture. And with that, we can have the fixation of a single haplotype in all of the population. Actually, you're going to have several evolution histories in each one of these subpopulations, and that is going to vary in the city. In Barcelos, we do have a population that uses preferentially natural breeding sites. So we have population in Pemixia, and you can have the fixation of a resistance gene. If it appears somewhere, it is going to spread throughout the whole population much faster than it would happen in Monsieur Lima. And for malaria transmission, this is also going to be interesting because just like the mosquitoes are structured in subpopulations, we believe that the plasmodium are also structured in subpopulation. And it is in the plasmodium that the mosquito reproduces, so you're going to have clusters of parasites in the city, which is what Marcelo shows in Monsieur Lima. In Barcelos, you're going to have a parasite that arrived there and is going to spread all over the city. They're not going to be concentrated in subpopulations. So both for control and transmission, understanding the structure of the population is very important. I have two more slides and I'm going to conclude. We did another study in Barcelos where we compared infected mosquitoes and non-infected mosquitoes. That was the master's thesis of Cinti. And we were able to find 8 locusts related, significantly related to infection. So here we have the frequency of these loci, the alleles of the infected mosquitoes, and here the control population, which is the rest of the mosquitoes, showing that there is a big difference. Some of these loci we decided to study in Anopheles gambi with the laboratory of George at Johns Hopkins. So we selected some of these loci. We found autologous in Anopheles gambi, and we analyzed to see if they had any relation with the infection. And one of these loci that we found here in chromosome 3, if I'm not mistaken, it's this locus here, showed us that when you inject mosquitoes with the interference RNA for this gene, you have a reduction in the number of non-infected and a reduction in the number of plasmodium of the mosquito significantly. We already had a paper written about infection comparing infected and non-infected mosquitoes with Maria Nisi in other regions of the Amazon, and we found other loci. So again, this diversity of the mosquito can be related to the diversity of its interaction with the parasites. So not necessarily what we found in a region is going to be what we will find in a different region. It had this evolution history of millions of years to then find the parasite. So when the parasite arrived in Brazil, it also found a huge diversity of Anopheles darlingi, and it has a big influence in transmission control. I'd like to thank Simone. All of the collection in Barcelos were carried out by her group and by herself. Marcelo for the collaboration in the project, and George from Johns Hopkins. Thank you very much.
Bom, chamo agora o professor
Now I
Paulo
invite Professor Paulo Pimenta from Fiocruz, Minas Gerais.
dispensa comentários, dispensa apresentações.
That needs no introduction, of course, needs no comment, but I would like to make a brief introduction and say that Professor Paulo studies the biology of the vector, the interaction of the parasite and the host, and the evaluation of strategies for blocking the transmission of malaria. His lecture has as the title The Control of Malaria Vectors, Histories, Perspectives, and Studies. Professor, thank you very much for your participation. Thank you. Thank you, Carol. Thank you, Danielle, for the opportunity to speak here. And as Paulo said, I'm not an entomologist. I am a control biologist. And my work is essentially an experimental laboratory work. We do not develop field research or work. I'm going to start by this slide mentioning a historical perspective of the study of malaria vectors in Brazil. We have to remember that Chagas already had a very important participation in the study in the beginning of the 20th century, showing that there is transmission of malaria inside the house. Adolfo Lutz was also very important because he was the one who established the concept of medical entomology, studying the biology of vector mosquitoes here in Brazil. And then a series of studies were published by Periaçu. These three researchers were very important for the study of medical entomology in the beginning of the 20th century. And in that beginning of the 20th century, it was very important to identify what we call the native neotropical vectors in 1926. So it's going to be 100 years this year that Anopheles dorlengi was described by Francis Metcalfe in the Baixada Fluminense in Rio de Janeiro. And then later, Anopheles equasalis was identified. I focus on these two because these are the two models that we work with the most when we study vectors here in Brazil. This slide is just to reinforce that there are many species of Anopheles.
We have many Danophylis species in Brazil. And the pointer is not working. It's here. So we have 400 Anopheles species recognized in Brazil and 60 possible vectors. We should restudy them to know if they are really vectors, but we should highlight that we should highlight that what we know today is related to this amount of vectors. I'm going to stay here. So those are the vectors we know in the Americas, the most well-known we have in Brazil. So it's interesting to understand that Darlingi uh, came down to this place, but now they are more located in the Amazon, as you know. So Darlingi, Aquasalis, and Albimanus are the most used in the lab in the experiments we perform. And it's interesting that when we started to work, we received uh, from Bento 25 years ago this Aquasalis, and we started working with Aquasalis. And why is that? Because we soon had this revision, and we used to say that an important problem we have was to establish the Plasmodium cycle in tropical regions. And today, it's possible for us to have many experiments infecting the vivax in endemic regions, and we work with Anopheles aquasalis, Anopheles albimanus, and Anopheles darlingi from the region of Manaus, a strain that we call RAS, Rosa Amélia Santana, who established this colony, the one that we studied the most. And at that point, we in this review concerning immune response. We had to work with microbiota, with genomes, and we put together this uh, malaria murine model. And we've advanced in all of these points. For the infection to happen in the vector, it should have this process. First, the forms of mosquitoes are the few ones that are able to send malaria to transmit malaria to humans. So they must bite, and then must cross the epithelium, and it must bite again the person. We have many barriers. The mosquito has ingested, and the first barrier is that the parasite must survive the midgut barrier, the digestive enzymes. They have to escape this escape barrier of the midgut, the midgut escape barrier, and then they have to go here and they end up in the salivary gland. Only after that it becomes a vector. And most certainly for these different stages, different species of parasites could face different problems. Besides the physical barriers, we also have immune barriers that prevent the development of the parasite. We developed some studies in the lab to see if the vectors were competent or not. And we used this system, uh, and first used that in my postdoc studies. We paid $50 for the glass professional to do this clone, and the blood is circulating with hot water around the funnel, and the mosquitoes are attracted to this food, and we use skin chicken skin or sometimes, and after this passage we dissect the mosquito to see the oocysts inside the mosquito, and we count them, and we see the spore sites starting to develop, and then we consider that this mosquito is competent as a vector, and we have this other stage because we see the presence of sporoids in this other stage. And then we had pioneer studies with Claudia, a doctoral uh, student, and we received aquasalis from Bento, and we got field mosquitoes because Darlingi was not colonized, and all of these mosquitoes infected with different infection rates. And here we have Aquasalis that was well infected compared with Darlingi. So we established Aquasalis as an excellent model to work in the lab, representing a new tropical individual, an Anopheles from the Amazon. And nowadays, more recently, we are able to see the comparison between Darlingi and Aquasalis. Aquasalis is infected with a lower prevalence, 64% compared to Darlingi, 80%. But when you observe the oocyst levels, it's very similar, and the mortality of infected people is only 20% in both. And here we have this high correlation with the numbers of oocysts and sporocytes. So for you to have an idea on how we solve this matter of working with both. So this slide is very brief, showing the the features of Anopheles darlingi and Anopheles aquasalis. And we have to observe the colony history. Aquasalis was colonized first. It's a gold standard in the lab. It's very important to work with both. The Halingi has a high susceptibility, susceptibility, and we see two colonies. We see the one by Maísa in the state of Rondônia, and ours uh, from our university, and and we started using them in several lab projects. And I'm going to talk about one that is very important. People asked us this question. People are going to be treated with the drugs. They go to the tropical disease hospital. They have this malaria diagnosis. So they go back home. What would happen to the mosquitoes that are going to bite these people? So we developed this work. Here you see the drugs used in Brazil, recommended by WHO, tefenoquine, primaquine, chloroquine, and artemisinin, and they are acting in different phases of the parasite in humans, and also it's we use these drugs in the foundation. And we have these three combinations: uh, artemisinin with primaquine, chloroquine with primaquine, and tefenoquine with chloroquine. So in the hospital, if there's a person that was diagnosed with malaria, they started signing some papers and they collected the blood in the first hours in the hospital. In the next hours, they went to the person's house to collect the blood. The person received the drug, has experienced treatment, and they're going to have an idea about the blood behavior concerning the mosquito. And then it was infected, and each one of the samples after 8 days and 15 days, we checked the sporozoites. And we saw that in all the combinations, that one extended the transmission time the most. This is a 72 hours. The chloroquine with tefenoquine maintains the mosquito infector, chloroquine and primaquine as well. And the treatments, they had this limited efficacy. So we must take action concerning that. I'm sorry that this is in Portuguese, but the anti-malarial drugs do not act on the first day of infection. How can we solve this question? So besides taking the drug, the person could take uh, ivermectin. We have two articles proving that. We have Anderson's and another person from Germany. So ivermectin is going to work in the mosquito that will bite this person under the effect of the drug, and the mosquito will die before the cycle is completed and before the mosquito is able to transmit. So, and also they could use repellents. Repellent is an immediate way to reduce human mosquito contact. And after this study, was an important one, we decided to investigate this question. We must have uh, this model of malaria in Brazil, the marine malaria study. And Alessandra went to Carolina's lab because she has uh, this very good model, Anopheles aquasalis, that is very good to use with this study. And she established that if you have treatment with pest trap from aquasalis, it's able to have this prevalence of 70%. So they infect this way, and post-infection we ascertained the presence of sporozoites, and showing that we have an aquasali model infecting a new tropical model, our model, with this marine malaria model that we could use. So that was a very important step for our study. Later on, we move on to to study Berge because Berge is much more used, and we used uh, Berge in Darlinge and Albimanas. And Breno surveyed several substances that were affecting the infection. We have the penstrap, uric acid, which is very important in some of the situations with the interaction. So using all these different substances that you see there, we found PABA. What is PABA? It's a pseudo B complex vitamin, and PABA was working very well in the case of Albimanus and also Darling, but Albimanus was the best case. You may see the closest amount compared to Darling, and this article that we just published was very important because we also saw Plasmodium berghei as a model, a new tropical model with this Amazon vector. This is a vector from Mexico in reality, not Brazilian. Albimanus from the Caribbean island. And we have both these models in the lab, working with Berge in the previous model that we infect with EOL. With EOL, that is used with many studies to understand the hepatic phase and the T cell responses, CD8 plus T cell responses, and it's crucial for vaccines aimed studies at preventing the disease before it reaches the bloodstream, and it's also used for studies of severe malaria and transmission, and it enables the formation of uh, and the use of chimeric parasites, the hybrid ones that were mentioned before. So you may use Plasmodium berghei to introduce proteins, genes, genes that will make them produce a surface protein of Plasmodium vivax, and you could test that for vaccines. And that protein that could prevent infection could be important as a candidate for clinical trials. That's why these two models are showing this huge advantage uh, to be developed in the lab. And there are other phases from the 2015 revision. We thought it was important to have the Aquasalis genome. We had one for Darlingi, and today we have Aquasalis as well. We found 12, almost 12,500 protein coding genes, and here we have annotated genes in putative functions uh, and with immune response. And that enabled us to have more assemblies and on immune genes, about the assembly of immune genes. Camilo is a Colombian student that is studying his doctoral studies with us, and we identified 278 proteins that are part of the immune response function, and we found those genes that are from conserved families. They are part of the defense pathways such as IMD, JAK-STAT, TOL. They are well known in Asian mosquitoes and African mosquitoes, especially in labs from Europe. And we're very happy because we are opening up this new pathway. And we also saw this work. We saw that the infection of our aquasalis with we have 244 differentially expressed genes, 209 are upregulated, and 35 are non-regulated. And now we are trying to better understand this study because it's important to have the expressed genes, and we can invest in new generation drugs, and we could work with next gen bioinsecticides. We could try to develop that in Brazil after this we have this kind of knowledge. And last, I'm going to present some details of what we are developing this last part. We started working with these mosquitoes, observing the farms, which are neurological modelers. Why did we choose the FARP neuropeptides? Because we in literature we saw that they are very important for the mosquito physiology, and they are neuromodulators. They are they are two related genes, the F and the short. The F is normal and the other is short. And here we work with cell biology, and we saw that this protein is secreted and it's present in the midgut, in the nerve cord, the salivary glands, and it's a candidate uh, to the work of insecticide that work in the neurological region. And the Berge is affecting and altering the FARP's expression. So basically, that's it. This is a slide that I love to show from our field work with the Foundation of Tropical Medicine. And that's what I wanted to talk to you. And these are the people working with us at Fiocruz, Oswaldo Cruz Foundation. We work with several institutions and several different people. And here we see Rosa's name. And we also worked a lot with Rita Day from Inca, who passed away some years ago. That's it. I'd like to thank you for your attention.
Vou pedir para os professores subirem para
I'll
a gente
ask
iniciar a
the
sessão.
professors to go back on stage so we can start the discussion.
Nós temos 10 minutos,
We
então
have 10 minutes,
para algumas perguntas.
so we have time for a few questions. I have a question to Ribola. I'll call you Ribola because there are two Paulos. That population, that structured population of Mancio that has subpopulations that are very different that you showed during your presentation, you mentioned the potential of the bird breeding sites having a role in this geographic isolation. In the sequences that you studied, did you look for the different feeding sources of these different populations? Maybe they're feeding off of different things. Great question, but we did not investigate that specifically. It doesn't have to do with food sources, but the interesting part is that when we get the adults from those locations, from those sites, we also collected the larvae from these sites. We collected adults, and 1 kilometer away, we collected the larvae as well. It is interesting that because using the genetics of the adults, almost 90% of the larvae we could identify as being from there. Or the other way around, because it's very structured. I believe that the main food source is going to be the people who live close by, but we did not check the food source specifically. If mosquitoes have breeding sites and food sources, they're going to establish themselves there.
Bom dia.
Good morning. Excellent presentations. I have a question to Ribola that also has to do with Bento's presentation. You observed populational structure in a small town, and the and the data from the paper that you discussed point out point to a very relevant genetic diversity of mosquitoes in South America when you compare it with Africa. Bento's presentation showed the Bento's presentation and studies showed the emergence of some populations in some cities, and in other cities they did not exist. So can we conclude that the emergence of resistance happens independently in the locations where it was observed, maybe due to the low genetic flow of these mosquitoes? Could you characterize that, uh, if you could characterize that there was not a lot of flow in a town, could we look at these towns where the resistance was detected and try to understand the processes that were applied to prevent the emergence of new resistance in the other locations?
Concordo com você, Rodrigo. Eu acho que o surgimento de resistência é
I
independente.
think the emergence of resistance is independent in each subpopulation in each city. We don't have flow between cities. If you don't have flow, if you move 1 kilometer away, you're not going to have any flow between cities. The problem is
Vai ser o mesmo surgimento de resistência em outras cidades com outro background.
trying to imagine if it's the same, if it's going to be the same emergence of resistance that happened in the previous city and the next city. There probably are a few points of resistance, a few resistance hubs, but I think it's difficult to estimate where it's going to come from with a different genetic background. But it is an important and interesting uh, analysis to make. Good morning. Thank you. Congratulations for your presentations. They were beautiful. My question is also to Ribola. I lived in Mâncio for 2 years. I apologize because I'm not sure if you showed the time frame of the collections, but I worked there in the area of endemics with the people who did vector control. And in those areas that you selected, the 3 points, historically, these areas had not undergone vector control for a while, for a few years. And there's a marker of sensitivity of the mosquitoes collected there. Not there, right? But this result that you achieved, wouldn't it be because of these mosquitoes did not have contact with any other kind of insecticide?
Eu não consegui detectar exatamente aquele
I could not detect exactly that
resistência do P450.
resistance point of P450, which doesn't mean that the populations are not resistant. We need to really study that, but if they're not under pressure, that could be an explanation as well. Good morning. Congratulations to the round table. I learned a lot. I have a question to Bento and to Alain. Alain mentioned the time, the hour, specific hour of the capture. In the 20th century, we participated in works with Machadinho, Cujubim, and other entomologists. We studied the immune response of the population of several locations in Rondônia, and they studied the entomology. That was a very vast work. They collected about 12,000 mosquitoes, and that they characterized from all different entomological parameters, and we used radiometric tests. Something that marked me a lot was how clear it was the curve. that showed the moment that the mosquito would bite. And that was very clear that it was at the end of the day. So when you mentioned midnight or 1 a.m., that was completely within the scope of what we saw. I would like to know if there was any change because you wouldn't expect to be bitten by a mosquito at noon. And for the preference, the feeding preference, uh, Darlinji had a preference for humans, but it wasn't 100% humans, of course. Not only human blood, but about 50%. And you showed 11%, and I was a little startled. I learned later that what we call um, malaria outbreak in the Northeast wasn't because of the Gambia. It was Arabiensis. Gambia is an excellent vector and as important as Darlinji, maybe. And this introduction of Gambia Arabensis actually came from Senegal. So, what is the importance of Gambia there? How did this confusion happen? When you talk about Gambia Africanos, are we sure that we're talking about Gambia? Maybe could it be another species in the Northeast?
Vamos lá. É assim, Cláudio. O que a gente observa em campo hoje é que a gente
Claudio,
realmente não tem um horário
what we observe in the field today is that there is no stipulated time. We always said that the mosquito would bite in the morning or at sunset. In some places, that is the case. I'm going to get the example of Porto Velho. We captured in Porto Velho 24 hours from 6 a.m. to 6 a.m. of the next day. In this case, we were working with Monsoni, but there was another another group that was working with Anopheles. We didn't see a lot of abundance in the morning, but when the sun set, we had peaks of this mosquito that were divergent in several areas. In Porto Velho, in the surroundings of Santo Antonio, we analyzed 17 points of collection with very little distance between them, and they were completely different, the emetophagic times in these points. For example, in a community, they would bite from 7 to 8 p.m., and a little bit further ahead, they would come up at midnight or 1 a.m. In terms of behavior, they acted the same, but they did not feed at the same time. That work that I showed you was done in a riverside area. It was a river that separates the municipality of Macapá and Santana about 17 kilometers away, and they were completely different. One mosquito would sleep when the other wouldn't, one had higher density and the other didn't. So, but the two towns showed cases of malaria. So we cannot say that it is this time or that time that they feed because that is not what happens in the field today. Probably it wasn't what happened in the past, but maybe they're more abundant during sunset. The past, the Arabienses or the Gambia were studied, but today I'm sure, and those who work with field entomology know that we don't have a fixed time for control. That is why the control activities need to be based on entomological data. If I choose, for example, to have a spatial application, a spray, if I choose to do a residual spraying, I have to choose a time when the mosquito is feeding, and with a high parity. The first mosquitoes are the new ones, are the newbies. They are clean. They never tasted blood before, so they are excited at 7 p.m. to sting someone. The older mosquitoes, they leave it for later because of the rush. People are more rested and they can get blood more easily. So we see that the mosquitoes that bite first, they are usually the younger ones. If you want to have true control, you have to do this analysis. We can't tackle this. You can tackle a problem if you're not sure of it. So the target has to be very certain so we can do the control. Maísa, first I would like to congratulate Alan for bringing this conversation about entomology. We truly have to bring this role of entomology for the control. The current tools, they have to be reviewed, and entomology has to take part in the studies. Bento, I would like to ask you a question and congratulate you for the initiative of FOG, which is an initiative that has been discussed and that the population prefers. It is very common in the Amazon region for them to ask for that. But there's a lot of lack of information about it. So my question has to do with your data that shows that there's no density modification, but you saw the presence of younger female mosquitoes. So my question is, did you follow this effect afterwards, both entomological effects, if there is an action or not, because it is a temporary work. You had 32 nights of collection during the action period of the fog. Very interesting question. Thank you very much, Maísa. Actually, this is a tool. It's what we call a fire extinguishing tool. And as Alan said, the analysis on the peak hour of the vector is fundamental. When we think about fog, we think about areas of mining where you cannot use uh, bed nets, but always associated to a treatment diagnosis. When we saw that the parity decreased, but the density did not decrease, we actually had a big discussion on that. Maybe we have to have two methodologies simultaneously, larvae and adults, because we're killing the adults, but the breeding site is giving birth to new mosquitoes. We're killing those who are in the environment, but there are new mosquitoes arriving every day. So if you associate the two methodologies, maybe we're going to have a better and more promising result. That is what we are discussing with Gilberto so that we can have a new methodology, and in these cases we can associate them. The fog is a fire extinguishing technique. It's when you have nothing else to do. Thank you, everyone. Thank you to the professors. And this is the wrap of our first table.